Hello, good evening. Okay, quickly I will revise what we have done so far. Nervous system and the coordination is the main lesson of this chapter. I think endocrine system we have learned previously. That is how hormone work in our body. And previous chapter we have learned ADH mechanism, and that's again same here: hypothalamus, pituitary, and kidney like organs, how it works with the endocrine system and hormones we learn. But what is new here is the brain and the nervous system, which is a bit complicated because there are so much of advanced mechanism happening in the nervous system. Okay, and we learn after that the basic unit of the nervous system. This is basic functional unit of the nervous system is the neuron. This is a typical neuron, and we can find uh, the long tail-like thing called axon, and the cell body, and the dendrites, and the synaptic knob. And there is a second group of cells. It's called a Schwann cell. So we will see what is the function of Schwann cell later. And there are three types of neurons: sensory neurons or motor neurons, and relay neuron or intermediate neuron. So these are the three types of neurons we have to learn. And then briefly we'll learn the reflex arc. That is how these three neurons join together to respond to a stimulus. Example, when we touch hot surface, the fingers are quickly withdrawn from this hot surface, which is example of reflex arc. And that is a spinal reflex arc because spinal cord is involved, but not the brain. I think this is the bit of advanced place now, resting membrane potentials. Here we learn how outside is positive and inside is negative of the cell membrane of neurons. This is called resting membrane potential. Then the neurons are at rest. There's a minus 70 milliwatt potential. And we learn these three transporters: sodium channel, sodium ion can come in; potassium channel, potassium can go out. This is continuously happening, but membrane still maintain outside high positive and inside the high negative charges. This is possible because sodium potassium pump. Sodium potassium pump. So 3 sodium pump outside and 2 sodium pump inside. 2 potassium pump inside, 2 sodium pump outside. It is active transport system, ATP is involved. As a result, when these three transporters operating at the same time, always outside sodium ion high, inside potassium ion high. And don't forget, compared to the other ions, when you take other ions, the net charge is outside is positive, inside is negative. That's a minus 70 milliwatt, and that's a resting membrane potential. Then we learn action potential, the rapid change of the membrane potential. Then again, we have additional two transporters, voltage gated sodium channel, voltage gated potassium channel. While these three are operating as resting membrane potential, if there is a stimulus, the stimulus open voltage gated sodium channel. Then the sodium ion moves in. This is rapid and large number of sodium ion move in. This goes outside negative and inside positive. Then at one point, this is closed, voltage gated sodium channel closed, and this open voltage gated potassium channel. Then potassium ion rapidly going out to make again outside positive, inside negative. So this kind of a rapid change is called action potential, and we learn this in another way. So I think this one always troubling is not drawing. Okay. And this is called depolarization. That is increased membrane potential to plus 40 milliwatt and followed by repolarization. Repolarization is again restoring the resting membrane potential. But still, there is a small change. This go up to minus 90. This is called hyperpolarization. This is due to prolonged opening of potassium channel. So time is 5 milliseconds to make one action potential. And this period, which is taken back to become resting membrane potential, is called refractory period. So refractory period also we learned last week. Yes. Right. So far, we learn these things. And if you have any questions, let me know. I think some of the notes not properly appear here, but in the recording, you can see rest of the notes for this page. Right now, today's lesson is propagation of action potential. So what is the meaning of propagation? Propagation is the moving the action potential along the nerve cells. Moving the action potential along the nerve cells. So if we take a electricity as an example, this is the bulb, and bulb is connected to the switch. So we have experience as soon as we switch on, immediately the bulb is light on. This is because the electrons can transmit through this copper wire, and this electron transmit energy. This energy source is converted into the light energy within a very few milliseconds, and we can see the light. So switch is off mean the electron flow is stopped. Page number 109. Page number is 109. Okay, now we're going to learn here how action potential transmit across the membrane. Right now, there are two type of neurons we can find. That means based on the absence or presence of Schwann cell. So first we're going to learn nerve which does not have Schwann cell. So what is Schwann cell? Schwann cell is a wrapping of the axons. Here, yes. So this is the axon. This axon is wrapped with another type of cells called Schwann cell. The gray color cells are called Schwann cell. So first we're going to learn how action potential transmit across the nerve. As I always told you, the direction is impulses this way. Impulse. Cell body to the synaptic knob. So this is always cell body to synaptic knob. So then they have to pass through this axon. So like this, they have to move to the end. So we're going to learn that how the action potential or impulses passing along the neurons. Okay, so I draw here axon. And I'm going to show the direction, this direction. Right now, we'll mark here so action potential number one is action potential is generated at position A. So this is the position A. Now the action potential is repolarization, depolarization, and then again come back to the resting membrane potential. So here the red color dot, there is a new action potential is generated due to a stimulus. Right? So that's a number one. Then number two, what's happened next? That's again axon. So I draw a dot here now. Right now you can see the same place, position A. Here's a position A. So it maintained now, it come back to the resting membrane potential. Resting membrane potential. So position A, in the second picture, it just finished everything. Action potential generate, depolarize, repolarize, resting membrane potential, back to resting membrane potential. So what's the outcome of this first action potential? So now it's a position B. At position A, now a potential is back to resting membrane potential. At position A, now potential is back to resting membrane potential. However, that action potential form new action potential at position B. Right now here I'm going to show again new action potential form at position B. So this one make a new action potential at position B. But when it make a new action potential at position B, the position A become now resting membrane potential. Right. Can you please copy this down? So number one is axon without Schwann cell. Resting membrane potential. If you have finished copying this note, just send me a small text done to the chat. You can send done. So I get the average time that you need to copy this one, then I can start. Okay, so third one, now third one, I will not going to show it here. Again, the position B become a resting membrane potential. And if I draw a position C here, then new action potential generate at position C. So it is continuing the event is repeating at the adjacent place that I will write C, right? The event is repeating at the adjacent place and continue along the neuron. Right, you can complete the note by writing the third step. The event is repeating at the adjacent place, example C, and continuing along the neuron. Okay, I hope you have written the sentence. Now, if we look again, action potential is generated on the neuron, and immediately that action potential diminish, become resting membrane potential. The action potential goes to make a new action potential at position B. So this event is repeating, and this is called impulses and propagation of action. That should be a spelling mistake here. Propagation. It's A. Right. Uh, Now, when we are learning some concept in nervous system, we feel this is not enough. Like, we can't understand this. We don't see something clearly, right? So we may miss some information. We need more information. I want to know how exactly action potential transmit along the neuron. So usually, if you want to find more information and answers or more explanation, certain areas you will never find because it is not available. That means we don't know. So we cannot find more than that. So then you must be wondering, okay, if we learn for advanced medical biology book, can we get a little bit more like things? So I will share with you. Okay. New share. Okay, so what you can do, you can type in Google how action potential propagate in neurons. So this website is called KenHub. What is KenHub? KenHub is a website for medical student, and we try to find if we can get more information. So this is something what we learn, the action potential, the graph. Here, propagation of action potential. Right. And action potential is generated in the body of the neuron and propagated through its axon. Propagation doesn't decrease or affect the quality of the action potential in any way, so that the target tissue get the same impulse no matter how far they are from neuronal body. The action potential generate at one spot of the cell membrane, right? So we learn this action potential generate at one spot of the cell membrane. We learn position A. It propagates along the membrane. It propagates along the membrane with every next part of the membrane being sequentially depolarized. That means sequentially depolarized mean one action potential form, adjacent place, again new action potential form. So this means that the action potential doesn't move, but rather cause a new action potential of the adjacent segment of the neuron membrane. So we need to emphasize that the action potential always propagate forward, never backward. So this is due to refractory period. So that's we just keep it away. So actually, this is the one discussed in this medical website. I mean, you can check also medical other websites and places. We cannot find more than that. So we always find only this. So one action potential form, then it become a resting membrane potential, and adjacent place new action potential form. So then that's how it's propagated. So this is axon without Schwann cell. Right now, what is the function of Schwann cell? So we write here, what is the function of Schwann cell? Before that, I'm going to draw you again the axon. Okay. This is axon, and this axon is wrapped with another cell like this. So this is a Schwann cell. Schwann cell is wrapped around its axon. So for example, if we look the cross section, that means we cut here and we look how it looks like, then you will see the axon like this, and around it, you can see the Schwann cell is wrapped like this. So something like this, the Schwann cell is wrapped around it. So that's the cross section, and that's how it looks like. Then there's an adjacent place, there's another one like this. And there's another one like this. Another one like this. And you can see a gap here. Always there's a gap. The gap is called node of Ranvier. Node of Ranvier. The gap is called node of Ranvier. Right, that is the Schwann cell. The function is insulation. So what is insulation? So it wrap around along the neurons axon, and it gives some insulation. That means insulation always trying to prevent some kind of an energy leak out. It could be heat energy insulation, or maybe electrical energy. It can also insulate. So this is due to a special protein and lipid in the in the Schwann cell. So Schwann cell has lipid and protein mixture called myelin. Myelin. So myelin is responsible for insulation. So that is a function of Schwann cell, and myelin is important to make the insulation. Schwann cell has lipid and protein mixture called myelin, which is responsible for insulation. Okay, you can also copy this one. If I still want to show the myelin, then I can color inside of the cells. And these are myelin. This blue color one is the axon. The gap is node of Ranvier. Right, you have to join again for the next session.